So good afternoon, everybody. Hopefully we'll end on a high. I think it's been a really productive and positive day. I'm always really interested to be at these sort of days where we get an idea of the bigger picture behind our industry. So just to give you a little bit of a background to where I'm based, I'm a lecturer in pharmaceutics in the School of Pharmacy in the Royal College of Surgeons, and we're in existence about 10 years. Of course, the original remit of setting up a new school of pharmacy was to produce more pharmacy graduates, and I was really interested to hear that point earlier on about clinically focused research. And I've worked in a number of jurisdictions, and I know in the most R&D active jurisdictions, the pharmacy community, schools of pharmacy, academics and pharmacy graduates are an integral part of R&D. So I think with the setup of additional schools of pharmacy, so ourselves and UCC in the last decade, that provides us with key personnel along with additional academic resources for industry. The other key component to the setup of the school was foresight on the part of some very, very eminent clinician scientists and biomedical scientists that are based in RCSI. So I think per medical faculty, if you like, across the country, we have the highest citation rates in clinical medicine. So obviously we are a single medical faculty, that's our specialisation. So we publish a lot of very, very high impact biomedical research. And about 10 years ago, they realised within the college that they needed two things. They needed to have the pharmaceutical expertise in-house to develop the molecules that were coming out of their labs and the understanding of the disease that was coming out of their labs, and also to increase interaction with industry. And that's where a pharmacy school very much fits into that piece. So therein lies the background. What they wanted us to do is bring what was being discovered in their labs, and we have world-class labs in all sorts of clinical areas, from respiratory to neurology to cardiovascular, and the structure of research within the college is based around that. And their key point was they needed to bring it to this part, and this was where we fitted in. So we got a state-of-the-art new facility built in St. Stephen's Green, so I could walk here today. How easy was that? So we're right in the centre of town, which actually makes interaction with other academic groups, other HEIs, and with industry, who are often coming in to meet the IMB on the other side of the green, very easy. In terms of what my own group focus on, we are a translational pharmaceutics group. So certainly at the start, when the lab was being formed, we focused particularly on the areas of basic biomedical research and applied clinical research. So we work with biomedical researchers. That's why I was really interested to follow Cormac's talk on trying to help them with understanding disease. So often they are now using difficult to deliver cargoes like nucleic acids at the understanding of disease stage. And we work with them in an area called molecular pharmaceutics, so delivering at a cellular level. And also, and I'll talk more about this, developing high throughput screening so that the biology is keeping up with the chemistry and what's coming out of those labs. With the clinician scientists, often they were hitting a roadblock at that preclinical testing stage. So not just to be thinking about the patient at the very end, but not losing really good therapeutic cargoes early in the development process. So before they went into in vivo animal testing, they needed us to intervene and comprehensively review what routes of administration they were using, doses, stability, and come up with a means of delivering them. And to focus on the areas that don't seem so cool in the biomedical efficacy focused labs, things like toxicology and PK and stability. So we work with them a lot. And then that morphed very naturally, and certainly it has increased logarithmically in the last 4 to 5 years, due in no small part to the cluster system in Ireland in terms of matchmaking industrial need, I think, with academic know-how into industrial research and commercialisation. So we develop our own IP, we work a lot on delivery platforms, and we patent around that area. But in a lot of cases, we're working with spin-outs, with SMEs, with large pharma on product development of new therapeutic molecules, and also with the med tech industry on device development. So we would find in a lot of the smaller device companies there may be a lot of engineering know-how, but there's not a lot of pharmaceutical development know-how, and we're very happy to step in there. So hopefully, college feel we've fulfilled our remit. So what I want to do, we work in a lot of different clinical areas, but a pet area of mine is a lifetime asthmatic. I'm currently undergoing my October wheeze. Pharmacist cure thyself is respiratory. So I have a very natural passion for respiratory research. And we have a very, very active respiratory clinical group in Beaumont Hospital, including both respiratory clinicians and biomedical scientists working in that area. And again, what was needed there to really translate those discoveries was a route of delivery. And for respiratory disease, we have a lovely natural targeting mechanism using inhalation. So inhaled medicines are around a long time. I know myself personally that they work. And what has happened in the last about 15 to 20 years is that the number of cargoes that we're interested in delivering via that route has increased. So there's an increased interest in targeting new respiratory molecules, so molecules to target local respiratory disease, and also the exubera story in terms of delivering biomolecules via the airways has increased. And what this did from a technological perspective is it really increased the research behind the appliance of the science, behind devices, behind formulation, along with the new molecules. So from the perspective of someone working in that field, the technology has really increased hugely in that time. So most people are familiar with a lot of the currently inhaled medicines. There's inhalers flying about all over the place. In terms of the classic examples are beta agonists and steroids for things like asthma, things like the mucolytics and Toby, the antibiotic for CF, more recently things like the antiproteases like alpha-1 antitrypsin. So primarily the focus is on local respiratory disease. But where the focus is moving is to growing what is already a huge respiratory prescription market and utilising new technology to grow the range of products that are on the shelves. And I always say to my undergrad students that the section in a dispensary that currently has inhalers is going to have increased dramatically in 10 years' time during their practice. So the global pulmonary drug delivery technologies market is predicted to be up around 44 billion by 2016. And a huge significant growth of that is going to be driven not just by devices, but what we put in the devices. So the material, the excipients, along with the biomolecule that we put in there. So a huge growing market. The kinds of molecules that we're interested in delivering are things like local molecules for targeting SLIPI, is another antiprotease, but like alpha-1 antitrypsin for COPD, and the systemically acting molecules. So certainly when we're looking at new routes of administration for biomolecules, trying to avoid the parenteral route, the airways is certainly one that are ones that we're going to see a lot of. So that's the potential. Those are the figures that are bandied about. In the real world, on the bench, when we're trying to do this, there's a number of very, very key challenges. And that goes right through from pharmaceutical and regulatory through to biopharmaceutical once it's in the body. So the big pharmaceutical issues are delivery in terms of efficiency. So when we were delivering the beta agonists, they were cheap, they were small molecules, we could afford for it to be inefficient and still work. We can't do that with biomolecules. So there's been a huge driver for new devices that can deliver lots of biomolecule into the lung effectively. The other big area is excipients, and there's a real dearth of excipients that are licensed for inhalation. We've got a very, very small list that we can choose from that are approved by the likes of the IMB or the FDA. And a lot of the reason behind that is poor screening tools. So we need to prove to the regulator that these excipients are okay. And that's an area we are doing a lot of work in. So that's getting the drug into the body. Once it's in the body, there's a huge problem with rapid clearance. Okay, so the lung was designed to make sure we're protected, and it does that in force. And also, particularly with some of the newer entities like the RNA molecules, the DNA molecules, there's very, very poor cellular targeting. So we don't have the means of doing that, and we need to have the means of doing that if we're really going to realise the full clinical potential of what's coming down the line. And last but not least, it was mentioned, I was really delighted to hear that earlier today, IVC. So having decent in vitro models to mimic what's going to happen in vivo. And respiratory has certainly lagged behind the oral world in that regard. So there's a lot of work that can be done, and by academics that can be done to support industry, I think, in that regard. So what I wanted to do was highlight where all of that research has now brought us in terms of how we interact with industry, and we do it a lot, and it makes my job much more interesting. It makes the students that I supervise, it makes their training far more relevant, and it makes them far more employable. So the drivers that I would see from the interaction that I've had to date with industry are manifold. It can be driven by therapeutic biomolecules that people have a need to deliver and are having problems with. It can be device companies, particularly inhaler device companies for respiratory. It can be a need to develop a delivery system for preclinical testing and models for preclinical testing. And last but not least, personnel training. So over 80% of the PhDs who graduate from my lab will end up in an R&D teams, generally within a very short space of time, leading them, I'm very proud to say. So they get a training during these sort of PhDs that is very, very relevant to the needs of industry at the other end. Their interaction with industry, I think, during that period helps enormously, both in terms of their understanding of pharmaceutical industry and also their networks, which are never to be underestimated. So what I wanted to do was sort of pick up on these three main points as examples of how we can support industry research. So one example in terms of a driver from a therapeutic biomolecule, this is this molecule SLIPI, which is very similar in terms of its efficacy profile to alpha-1 antitrypsin, which is now licensed. And SLIPI was of enormous clinical interest. There were clinical trials done. And the efficacy side of things seemed to be there was a very strong case there in terms of it's a very, very strong antiprotease, which helps in the inflamed lung. It also had additional anti-inflammatory, antibacterial activity. But when a patient inhaled this drug, it was cleared incredibly rapidly. Not only that, as it passed through the inhaler, it was degraded. So this was a molecule that was thrown my way to see if I could come up with some means of improving those problems. And the strategy we came up with was a tried and trusted method in pharmaceutics in terms of liposome encapsulation. So what we did was we used a particle engineering approach to this. The key, for those not familiar with respiratory drug delivery, the key in delivering anything to an anatomy as complex as the airways is particle size. So we have to be very, very careful in designing an inhaled product that is within the right particle size. So the key parts here are your therapeutic, the excipient, the material that you use, and the process that you use to create it. And therein, you get your particle that's fit for purpose. I mentioned the material component of that. In this case, we used liposomes. There's products that are using these in the market. We often try and start with a material that's already licensed for administration via other routes to keep our regulatory hurdles lower. But these are limited in what they can do in terms of what we need them to do in the lungs, and certainly there's a huge area there of need around more advanced materials, I think, for airway delivery. So what we were delighted to see was that we were very quickly able to control this clearance of SLIPI in the lungs. So this is an in vitro system in an airway cell model. This is the SLIPI alone, and it's transporting across an airway monolayer really rapidly. We encapsulate it in a liposome and it slows it down. We went to in vivo, into an asthma model, and we could see the free SLIPI appears in the bloodstream really rapidly. We encapsulated it into a liposome, and very, very little was appearing in the bloodstream. So being the academics that we are, we wanted to find out where it went. Equally so, industrial collaborators. So where is it going if it's not going into the bloodstream? And what we found was it was being picked up by particular cell types within the lung. So we had serendipitously fallen on a targeting system. And we went back then, so we went back from in vivo in an unusual study to in vitro. And using high throughput, high-tech epifluorescence microscopy, we were able to look at the cell types. So this is our targeted system. We're able to see lots of red dots represent our drug molecules being taken up. And we were also able to tailor that and get quantitative data for that. So we have some of the best fluorescence imaging kit in the world in Ireland, thanks to huge amounts of funding from the HEA and SFI. There's a huge amount of toolkits there. We're applying it there for airway cells. You can apply this kind of technology for a huge range of cell types depending on your clinical need. And we went on then very quickly just to say we went on to develop that system then as a dry powder inhaler, because for biomolecules and particularly for stability reasons, it's beneficial to have a dry powder inhaler. So that was just an example of the kind of therapeutics. There's a lot of small companies now approaching us with small molecules and biomolecules that they want to inhale and they're coming to us to do early stage work on toxicology, stability, formulation. So I am seeing a real need there from both small and large companies in that area. The example that David mentioned was Aerogen, and this has been a fantastic, the easiest collaboration I've ever been involved with, academic or otherwise. It's a win-win from both sides. We work in the area of respiratory delivery, we've got lots of biomolecules, but we were not engineers and we did not want to have to design a new device. Aerogen had gone and put all the hard work into developing a really lovely air and neb vibrating mesh technology that delivers biomolecules really nicely. But what they don't have is pharmaceutical know-how. So as part of the IDDN, we worked with them on a number of different projects, looking at different things like optimizing the performance of the device, expanding the applications they're using it for, and finally adding some value with new products. So in terms of optimizing performance, they knew this device worked pretty well with cargoes they were putting through it. What they hadn't looked at was the fluid and the properties of the fluid that were being put through it in huge detail. So we went back to the lab to basics. We looked at properties like viscosity and surface tension. Very, very simple studies to do, but a bit of know-how required to do them properly. We went back and did this, and we looked at how that affected the performance of their device. And I know now they go into meetings with big pharma and with other companies who are looking to use their technology, and they use this information in terms of their pitch to them and to be able to expand their collaborations. Because if someone's coming to them with a particular system, they know how it's going to perform because we have provided them with that kind of data. David talked about this already, so I won't go into it in huge detail. Another thing they were really interested in was expanding the applications. Because we work with so many different clinical groups who had interest in lots of different therapeutic cargoes, we had access to a lot of different therapeutic molecules and the know-how how to characterise them. So we did a lot of this work on a number of different cargoes. An example here is calcitonin, and this again helps them in scoping and broadening the applications of their technology. And last but not least, I mentioned this already, classical inhaled therapies. A small molecule is inhaled into the lung, it sat on the surface of the cell, interacted with the receptor, did what it needed to do. Newer molecules, particularly like the RNAis, and there's a huge growing family of these in terms of microRNAs and shRNAs, need to get inside the cell. So the RNA can get them as far as the surface of the cell. Then we have to have some means of getting them inside. And we work on putting nanoparticle device technologies together to create a platform that can do that. This is just some data from this system. The key one here is we were able to nebulize these nanoparticles onto a differentiated Calu-3 culture, so airway culture, and we were able to see knockdown, because one of the key things here is when you nebulize a nanoparticle, it can destroy everything that's being put through it, depending on what that material is. So a very promising platform that, again, Aerogen and ourselves can provide to biomedical, clinical, other industrial partners that need it. So last but not least, preclinical testing, and this goes back a lot to what we talked about earlier today in terms of regulatory. There's a huge and growing need to be able to properly test new molecules and new materials. And we've harnessed all of the wonderful epifluorescence microscopy that's available now in Ireland to be able to screen nanomedicines in respiratory cells. So this epifluorescence allows us to look at nanomedicine uptake at a cellular level. We can quantify it, we can get really pretty pictures, and we can track the particokinetics, so we can check the kinetics at a cellular level. It also helps us screen novel biomaterials, and we've had a lot of matchmaking with us and biomaterial scientists interested in screening for different routes of administration. And last but not least, what we're really interested in at the moment is harnessing another one of Ireland's great strengths, which is bioengineering. So we have a really good background in this. We've got a really good background in drug development in Ireland, and we're trying to put the two together to make Ireland a leader in 3D modelling of different tissues. So instead of the molecules we're seeing lost in translation in preclinical rodent models, can we somehow use human cells? And this, I know the FDA are really keen on this direction for drug development. Can we use human tissue models to test drugs, both for disease models and for toxicity and immunogenicity? And that's a growing area of interest for us in the group. So I'll finish on a high note before I pass over and before everyone keels over, because they're waiting for their sweets from Trick or Treat. We had we had a meeting, that's why I picked respiratory specifically for today. There was a meeting of most of those of us in Ireland who work in the respiratory field, from clinicians, biomedical scientists, delivery scientists and industry, trying to put us all in one room to see where we had skill sets that could help each other. And certainly I think there's probably a real niche there in Ireland for getting those sorts of clinically focused groups together and all talking together to see how we can help each other out even more. We're doing it well in respiratory, we could do it better. The big thing, the big take home I have from my own experience, even within my own college and within the network, is there's a huge amount of fantastic basic biomedical research being done. We've seen examples of it today. We will not see that translated commercially or clinically unless people are provided with the tools to do the preclinical testing in the first instance properly, and then to be able to scale up and manufacture. So hopefully I've made the science of pharmaceutics sound indispensable. Last but not least, to acknowledge all of the funders who have funded research to date. This is the list of just my respiratory collaborators. The ones in the other clinical indications would be too many to list. And just as David mentioned, the work we've done has helped us leverage non-exchequer funding, which is really important too. So thanks, everybody.